Hello, my name is Brendan Detterman, currently a senior here at Oklahoma State University, and my research is over the pulmonary dendritic cell subset interactions with Cryptococcus neoformans. So what is Cryptococcus neoformans actually is? Cryptococcus neoformans is an opportunistic fungal pathogen that can typically be seen in dead or decaying tree bark, or sometimes pigeon glotto, via inhalation that can cause cryptococcal meningitis. Immunocompromised patients who are suffering from anything that can lower their immune system, such as HIV or AIDS, are at great risk, especially for those in sub-Saharan parts of Africa. Cryptococcus neoformans is thought to survive intracellularly and be killed by innate cells, but if that doesn't work, they're actually able to replicate at great numbers and escape and evade these innate cells. Prior to this research, there were other research labs that found two subsets of cells, those being dendritic cells, and of course those being CD11Bs and CD103s, capable of interacting with Cryptococcus neoformans. My research hypothesizes that these two different subsets of dendritic cells are actually able to show different interactions with the Cryptococcus neoformans being incubated with it. In figure 1 is a depiction of how we're actually able to separate and purify those cells. Using a CD45 antibody marker, we insert this through the tail vein of a mouse using a mice model, and we're actually able to only collect cells from the pulmonary section of the lungs of these mice. Next, we want to be able to positively and negatively select for the cells that we're actually looking for. As you can see here in figure 1, we are positively selecting for CD103 positives for the CD103 positives and negatively CD103s for the CD11Bs. Figure 2 is a depiction of how we're able to actually quantify and see how much our cells are paired and how well our cells are paired. A full cytometric analysis allows us to see that, and we're able to see that even though these graphs are kind of confusing, we're able to know that we're gating against a control of positive and negative selections. We're able to quantify how much of our CD11Bs and CD103s we actually have, and there, of course, nothing else in those population samples. Figure 3 shows an antifungal activity as well as an association of our dendritic cells with Cryptococcus neoformans. Figure A shows male and female mice and their uptake with Cryptococcus neoformans. Uptake meaning these cells are actually engulfing Cryptococcus neoformans and they're actually associating with Cryptococcus inside of it. As you can see in figure A, both male and female are interacting pretty well with the fungal pathogen. In figure B, we wanted to observe antifungal activity. As you can see in the males, there shows a significant difference of the antifungal activity between the CD11Bs and the CD103s. The question that was posed to us after this discovery was, if they're not killing Cryptococcus neoformans, what are they actually doing? As you can see in figure B, for females, there is no antifungal activity for both either subset. Figure 4 allows us to see the cytokines that are recruited and the chemokines that are produced during a cryptococcal infection. The solid lines show our subsets without Cryptococcus, and the dashed lines show our subsets with Cryptococcus. Figure 4 is a representation of all the cytokines and chemokines that are produced, but there is no significant or statistical analysis that shows significant evidence of anything being inhibited or produced greatly. Figure 5 shows the intracellular morphology of our pulmonary dendritic cells with Cryptococcus neoformans. One of the things that I want you to take away is knowing that these little white dots that our M. cherry strain Cryptococcus neoformans possesses is that we're able to see our Cryptococcus strain under different channels under a different dye that is, of course, how it is, stained. One thing I want you to take note of is how if Cryptococcus is, of course, condensed or shows a C shape, that means that the dendritic cell is actually able to kill the Cryptococcus neoformans, and that's something that we want to see. However, if you see a round, nice shape, such as right here for the male dendritic cells of the CD11Bs, that shows that the dendritic cell has not successfully killed Cryptococcus neoformans, and now we're wondering why that is, of course, happening. The big takeaway from figure 5 is showing that is showing the antifungal activity representation of figure 5 and figure 3 comparatively. Shows that how both dendritic cell subsets are not inhibiting the cryptococcal fungal pathogen. In conclusion, we found that both subsets of CD11Bs and CD103s are associating pretty well with the cryptococcal strain, as well as knowing that knowing that the intracellular morphology of these cells are showing depictions of the cryptococcus inside these dendritic cells. Our future directions lead us into our research of hopefully finding an RNA sequencing as well as a gene study to see what genes are actually being turned on for chemokines and cytokines during the cryptococcal infection. Thank you for watching.